People have been occasionally asking me why do we have this conference in Dublin, and of course the real reason is that we don't have to pay the travel expenses for our next speaker.
I'm very expensive. Let's just see. So while, of course, I'm also expensive and have caused a bit of nightmare because I'm going to use my own laptop. And what I'm going to do today is talk to you about the most extraordinary and wonderful of all mammals. And these are, of course, the bats. Now, this session today is called Neglected Species. But bats are also some of the most vilified mammals of all times. Now, I'm asking a question because I was told at the talks, or at the gala dinner last night, so much more fun for audience participation. What's that? What is that on the screen? What animal is that? Anybody? It's a bat, you're very good. But it's actually a vampire bat. There are 3 species of vampire bats in the world, and that's one of them. And I want to read a quote from Bram Stoker. To instill fear into the heart, I became a bat, a monster in the night. Well, actually, that's Batman, not Bram Stoker. But Dracula was written actually in Dublin, only about 20-minute drive from here. But bats, yes, they make people scared. But what I want to do today is I want to tell you about the work that I started about 12 years ago, based on the inspirational research of Steve Ousted and also Vera Gobernova and Andre, about why we should study bats. Bats are uniquely long-lived. Bats have evolved mechanisms to somehow slow down the expected aging process, given their body size. And so by studying bats, we can find ways to live healthier longer. And address, of course, one of the greatest challenges that society faces today is to find ways to extend our health span and ultimately our lifespan. But there's something else that bats do that we haven't talked about. Bats are able to live with and tolerate many diseases that make us all sick. And the second grand challenge in the world, and we know now because of SARS-CoV-2, life expectancy in the US dropped because of an infectious disease. We need to find different novel ways to fight infectious diseases. And I'm going to argue and show you that bats have evolved adaptations that can allow us to solve both problems. Therefore, studying the most vilified creatures of them all holds solutions that we deeply need. So again, this is inspiration from Steve Ousted's 2010 paper. To me, it was a seminal paper. Again, there's your longevity ratio. You can predict how long things will live for given their body size. As you've heard, whales, the largest of all mammals, living over 200 years. You'd expect that. But bats are the smallest of all mammals. The bumblebee bat is the size of a little bumblebee, and they can live for an extraordinarily long time. And if you were to roughly equate body size and say, well, how many human years can bats live for? The longest-lived bat is Myotis brandtii, caught in Siberia. As an adult, tagged, released, was caught again, I think it's now 43 years later. That individual, that species, weighs 1/3 of a lab mouse. So that's the equivalent, roughly, give or take, of 280 human years. Living in the wild, no protection, finding ways to survive, finding ways to live long, live healthily, little to no signs of aging. But it's not just one species. The majority of the 1,500 species of bats that we have studied in a marker capture way can live for a long time. But again, they're resistant to many of these pathogens. And the question is, are they really? Yes. You can inject bats with SARS. They don't get sick. With rabies, sometimes with Ebola. You could do tests in cells, and they seem resistant to it. So what have they evolved? And because the reality is, even if your biological age is 20 and your chronological age is 70, you're doing real well. But if you get an infection and you can't resolve the infection or resolve your ability to have a highly inflammatory response to that, you're going to die. So we need to find ways to live healthier longer, and studying bats are going to help us. And I want to tell you a little bit about this quest, this journey I've been on. How do you use bats as alternative model systems to uncover the molecular pathways that underlie their extraordinary aging and disease tolerance in mammals. And I want to pitch this idea to you of what have we done and what do we need to do. We have to ask the questions, how do bats defy this aging process and limit disease? But there's kind of a catch to model bat. The catch is they're wild, long-lived mammals. The longest-lived bats, you cannot keep them in captivity. Our husbandry is no good. So therefore, we need to study them in the wild. How do you age a bat? You have to catch it as a baby. You have to put on a transponder or a tag, release it, and recatch it again. So this means you need decades-long microcapture studies. They're not nice little things like butterflies or bees. No, no, no. They come out at night when we want to sleep. They find resistant species will actually bite you if you want. So you have to. It's difficult. It takes decades long of study, but we need to do this. So the two questions you have to ask is, well, what have bats evolved to allow them to resist disease, to control viruses, and to control their own level of inflammation? What about damage tolerance? How can they avoid the damage of living, given that they fly, really high metabolic rate? How have they evolved to be able to tolerate this damage? And so these are the kind of areas I think we need to look at. These are the areas that we've shown in the past that there's something different going on in bats. This is what we're going to argue leads to the exceptional longevity. We're going to need to study wild populations right across the aging spectrum. You have bats that are extremely long-lived and bats that live for as long as would be expected given their body size. Let's look at them and see what's the difference. We're going to need captive colonies because you cannot lethally sample your bats in the wild. They're all protected. So there are some excellent captive colonies around the world. Typically, they're the fruit-feeding bats. But where we'll be able to cull these bats, we'll be able to have enough tissues to be able to do these experiments required, to be able to then go to look at this non-invasive sampling we have to do in the wild and make it meaningful. We need comparative genomics. We've got to sequence those genomes to exquisite level assemblies to uncover the things bats have evolved. But we also need to validate this. You can't just say, oh, that's a lovely correlation in the wild and in captivity. We have to find ways to bring the bats into the labs. We need cell culture. We need bat mice. We need to move this forward. We have to then say, right, we have a regulator we think drives the aging process. We need to validate it in something. Potentially, wonderful C. elegans that have these 10-fold different changes in longevity. Could we stick in bat genes into C. elegans and make them live longer? Then, of course, we're going to have to make bat mice. Bat mice. But which one of all these different changes in bats are we going to go knock into a mouse and see will it live longer? So that's where those worms come in, to help us try and fine scale and pick our targets. Hopefully we'll understand how we can have increased disease tolerance and health span extensions. So bats are going to give us this biological insight. We're going to learn much more about the aging process in different species. And I'm going to argue we're going to be able to make drugs. You saw how AI can help find particles, molecules, and so forth. Everything's moving more quickly. So this is how I think we do it. But does it work? What have we seen so far? So I'm going to tell you the tale of where we're at the moment. So first of all, let's talk about our microcapture projects. So for the past 11 years, I've been working with Fred Touzelan and Brittany Vivant in France, whereby we've been studying this long-lived population of Myotis bats. These are the longest-lived genera of all bats. We go out to France every year, we catch all the entire colony, we mark all the babies, we recapture them. We take a teeny bit of blood, we take some wing punches, and so we have now over 6,000 known age specimens taken from individuals that age year after year after year. This is great. But these are the longest-lived genera. We also need to try and look at species of bats that have different longevity and different LQs. And so we picked these 4 different species. We have our Myotis myotis, the highest LQ. We have Rhinolopus furbequinum, beautiful horseshoe bats found in the UK. A 60-year microcapture project that we work with. We're doing the same thing. We're looking at captive bats. These are the Rosetus aegyptiatus. These are kind of middle-aged bats. And then we're looking at molossus. And this is the shortest-lived bat, given its body size. It lives as long as expected. And it's found in Panama. And just to show you what fieldwork looks like, A lot of this work is done by my lab. They're having a great laugh. And see the fella in blue the whole time? That's Ty Glonigan, who's been really, leading this. He right now is in Bristol sampling these horseshoe bats. They have to go to Panama, Copenhagen, Bristol, France, lots of places. So you got to do lots of field work if you're in my lab. Also, we need genomes. And so I established with Sonia Vernet's Bat1K, this global sequencing consortium. We're dedicated to sequencing the genome of every single living bat species. We've pushed forward the methodology we use to sequence non-model, non-human organisms. We're at phase 1, where we're sequencing a representative of all the families of bats. We have 113 reference quality genomes sequenced at the moment that we're working on. And I'm going to tell you where we got the funding from this. It's an interesting biotech story. Well, what about lab validation and functional analysis? We're moving on this. We're now able to take wing punches and grow fibroblasts in our lab. But what about bat mice? I'm going to tell you the tale of one little bat mouse at the end, and does it work? But working with Thomas Zawicka, and again, we need to work with collaborators. We found a way and developed a methodology where you can take a 3-millimeter wing punch from any species of bat, use this new batified Yamayaka process, and make iPSs. And we've been able to differentiate those into different blood cells. So we're starting to move forward. Because remember before we started doing all of this, there were no tools to be able to make bats model organisms. But what did we find? So I'm going to tell you the story of a couple of our markers and bring you up to date where we're at with some of these studies. It's a little raw because it's just hot off the press. But let's look at telomeres. TTA, GGG. The repeats on the ends of our chromosomes, that every time our cell replicates, they get shorter and shorter and shorter, unless they're the germ cells where telomerase is there. What happens with bats? Well, in a previous study, where we were studying bats and aging over known age individuals over 6 years of meiosis, what we found was that in the longest-lived bat species, their telomeres did not shorten. No matter what we did, we couldn't make them shorten with age when we did our statistical analysis. We couldn't find any evidence that bats were using telomerase to maintain their telomeres with age. So what do we find? So as we kept this study going, now it's 11 years, we've got the much older individuals, we even have a 14-plus young lady. What we found that in meiosis, in the longest-lived genera of bats, still, we do not find a shortening of telomeres with age. So somehow, the longest-lived bats, their telomeres don't shorten. As we start moving down the LQ, these are these Rhinolophus firmicutes. As you can see, we have a lot of the older individuals. There is a shortening, but not quite as steep a slope as would be expected as other mammals. So we're looking at that. We have over 1,800 individuals here in the study. This is Rosetus egyptiatus. These are these captive fruit bats in Copenhagen Zoo that are culled because they want to maintain the size of their colony to keep their welfare well. And we're able to sample them. But it looks like in these captive bats, and captivity may be a problem, but that you do find this weird shortening. So something's different going across bats. But here's molossus. I'm very proud of this one because we found that if you look here at the age of 0, 1, 2, 3, 4, 5, we have a couple of 5-year-olds. This has wrecked everything. Nature wrecks things. They're not supposed to live that long. But yet with our marker capture project, we found they were living longer. But you still, when you look at the survival curve, you know that typically they're not most likely going to live past 5. We're still doing this work. But telomeres do shorten in the shortest lived bat. So what's interesting is, as you see, meiosis, Rosetus, Rhinolophus, molossus, the rate of shortening seems to correlate with their LQ, and that's interesting. But how are they doing this? So we have our genome sequenced, as I told you before, reference quality genomes. And you have younger and shorter-lived bats that are sister taxa. And I'm a phylogeneticist. I'm really an evolutionary biologist. And what you want to try and see is when you look at genes that are involved in maintaining telomeres, you sequence them, and you see are they evolving differently in the short-lived versus long-lived bats and vice versa. They're closely related. Does anything hop up? And it does. This was actually very interesting. In all of the shortest lived lineages, in comparison with their closest relative, what we found that there was evidence of selection. Something was different, but on the same gene. It's called phylogenetic independent contrast. It's a very powerful way of helping find regulators. And it looks like it's this ORTEL1. It's been shown to be involved in multiple diseases. So this is a target that we really need to look at. You find when you look at the expression of the genes taken in blood, so we'll sequence the entire blood transcriptome from young, middle-aged, and older bats. We look at meiotis, and here's looking at molossus, the shorter-lived bat. And you find actually there's a difference in the expression of these sheltering genes between the youngest and the shortest lived. But this is only n equals 16 of molossus, so we still need to work on that. And we haven't got the oldest one sequenced yet in the analysis. But there still is a difference. But look, TERT's not being expressed. Doesn't look like it's telomerase. What's going on? As Vera brilliantly showed you, RNA, and I'm quite horrified, mightn't actually be the full picture. And this is work done in collaboration with Vera, with Andre, and with Fatima, a postdoc in their lab. We sent the cells there and said, why do you look for the proteins? Do a trap assay. And what you see when you see this, there's meiotis and rosettis. Darker in black means more telomerase. Lighter means less. So it looks like telomerase activity is actually happening in these primary wing punch fibroblasts that we grew and sent to Vera's lab. Well, let's think about this. They have telomerase, but there's little to no evidence of cancer. How are the bats doing it? Do they have immune regulation? Do they have a better way of regulating their cell cycle? We need to look at that. And when are they doing it? And again, with those horseshoe bats, these Rhinolophus firmicutes, we now have evidence of how the telomeres are changing within short time frames. And it looks like during hibernation, bats go into hibernation in temperate climates, there is an expression of telomerase. Well, not as the expression. Their telomeres are growing, you can see here. And a recent study that came out in China, where they took wing punches, sequenced the transcriptome during hibernation, we found evidence for increase of telomerase. So maybe they are using telomerase. Maybe you can't see it properly in the blood, but you see it in the wings. And again, we need to be able to bring together genomes, transcriptomes, proteomes to answer this question. Fibroblasts. This picture of Orlin Alcock working in my lab, where we're able to take these wing punches, bring them back into the lab, grow out primary cultures. We want to say, right, how good are bats? How resistant are they to different stresses? Let's try and do it in the lab. Again, to remind ourselves, we have right groups, which are mice. We're looking at the short, the medium length, and the longer-lived bats. Now, what would be our expectation? When we stress the cells, we would expect the longest-lived bat to be more resistant, right? More resilience, we'd expect that. When we looked, we use this MTT assay. Basically, as you can see here, this is hydrogen peroxide, nasty thing for cells. The more you add, the less viable the cells come. So this was the assay that was being run by Orlin in my lab. So what do we see? When we look at cadmium, a DNA stressor, what we actually find is that the longest-lived bat is doing the best, can put up with more and more stress. That's what we expected. Mice not doing so well. Rosettis kind of doing a little bit better, but meiotis, by far, seems to be able to deal with this. And we've shown from our transcriptome analysis, recapitulating what Vera said, DNA repair is very, very important and increase with age in these bats. We put in molossus. Uh-oh, what's going on? She's not supposed to be there. The shortest-lived bat's not supposed to be doing that. So nature is complicated. We're trying to understand what does this actually mean. We looked at tunicamycin. Again, I would have made the assumption that bats would be much better able to resist tunicamycin stress, which a proteasome stressor, than mice. But it's the other way around. We throw in our long-lived bat, our shortest-lived bat, it's doing something very, very different. What's going on? So what we'll find, just to summarize a little bit as a result, so the longest-lived bat, much better able to resist these DNA stressors. With the tunicamycin, a proteasome stressor, not doing so well. But I know that in Vera and Andre's lab, they've shown that this actually is the same in these long-lived rodents. When you compare the longest-lived ones versus the shortest-lived ones, they're not that good at dealing with tunicamycin as a stressor. But this is just a way to show you how we got to move forward. What about mitochondria? And I haven't really talked about these before. What we found when we looked at heteroplasmy with age in these bats is that they do not show the expected level of damage to their mitochondria given their high oxidative rate. But what's going on? This is work that was run by Ilse Skogina and Sebastian Riquier in my lab. And so what we wanted to do is we wanted to focus on this blood transcriptome. A little bit of blood, sequence all of the genes, look at the genes that they have differential gene expression with age, ones that are upregulated and ones that are downregulated. And we wanted to focus on these mitogenes, genes in the mitochondria and the nuclear coding genes that manage them. We want to see how does it change. And these are in the meiosis. Meiosis is our biggest transcriptome study at the moment. We found a whole series of genes that actually are downregulated with age. You can see them here. Involve aerobic ATP production, mitoribosomes, and beta oxidation. These are these exclusively downregulated pathways: energy metabolism, neuromodulation, liver disease, cancer, body temperature. These guys are going down in bats as they're aging. Look at the 10 most upregulated genes. Again, ATP pathways, cell scavenging, cellular signaling, and so forth. Ilse is very interested in these aerobic, these muscle fiber twitches, so she can talk to you about that. These are these exclusively uprated pathways. Autophagy is a big thing, the ability to scavenge and remove damage. Is that why they're not showing the expected level of heteroplasmy? But this is what I find really interesting. When you go and you look at genes that are upregulated, downregulated, or pathways up and downregulated, you're going to find that they're pathways that are doing both. So you're finding genes in pathways that are both being upregulated and downregulated. Means that bats have evolved ways to tweak the regulation of those pathways. And these pathways are particularly involved in neuronal diseases, heart diseases, Parkinson's, ALS, Alzheimer's, Huntington's, diabetics. So these are inflammatory diseases. Bats, by modulating the control and regulation of their mitochondria, have found different ways. So these are good avenues to explore. What about more functional validations? What about this little cutie here? What about a bat mouse? Is it really impossible? So work done with my collaborator, Lim Fu Wang's group. The genomes were sequenced, and we asked the question, well, what is going on in bats' immune genes? They're inflammatory genes. They're different than other mammals. Bats are missing the entire suite of these pain genes, these inflammation genes, that every other mammal, including us, have. They mount a very different immune response. One particular gene is ASK2. And ASK2 is an inflammation gene that shows a different base pair constitution in bats. So Lim Fu Wang's group knocked in a bat ASK2 into mice, the proper knock-in mouse, and challenged these mice with the flu, with lots and lots of other different stressors, and found that when you infect the mice with the flu, if they have the bat ASK in them, they live so much longer. But what's really tantalizing is not only can they survive disease, so their inflammations drop down, it looks like if you don't cull those mice, they have much better health span markers. So again, inflammation potentially is at the heart of bats being able to tolerate pathogens because they limit their cytokine storm and also able to limit their age-induced inflammation. So you can do it. But how do we do all of this? We need funding. So a lot of what I've showed you is a potential grant application, and we need to be more cautious in our grant applications. But thankfully, people have started to listen. And investors have thought, well, actually, working with bats is a good idea. And I wanted to point to this company, Paratus. So Paratus Sciences is a new startup company. There's a $100 million investment into this company. Amir Nashat was really one of the founding venture capitalists and getting venture capitalists involved in this. But the idea is that this company wants to study the unique adaptations that bats have evolved to make therapeutics for the future, particularly looking at how we can deal with and mitigate inflammatory disease. What's really cool about this, that it's a little outside the box. So I'm one of the founding SAB members and have been a consultant since it started. But part of it is I said, well, if I want to get involved, and if you all want to work together, we need to make sure that, yes, we can sequence these bat genomes. So primarily, Paratus is sequencing the 1,400 bat genomes. but in collaboration with Bat1K, this genome sequencing consortium that I'm one of the founding members of. But this is released to the public. And so this is a very, very unique synergy of scientists, the public, with venture capitalists, with biotech, to be able to move and change the world, to bring it forward. Our genomes are being released right now as genome notes. There is no proprietary, I suppose, take on the genomes. The genomes have to be released. We're all working together so far. I think it's an extraordinary, extraordinary show of how synergy can actually move things forward. But I hope I've convinced you that studying these bats will allow us uncover exceptional longevity. And it's an interlink between how you can tolerate the damage of living and tolerate the damage of disease, and the two go together. And that really studying bats will give us much more insight into the biological process of aging, new targets for drugs. But I want to show, well, if I haven't wrecked everything, this. So this, by the way, we're going to give her a name. We're going to call her Eve. So this bat, she was caught as a baby. She was caught as an adult in 2010. She had given birth. She had a tag, a transponder put on her. We've caught her again now, 2023. She is 14 plus. She could be 30. No signs of aging, great weight, lovely fur, had just also had a baby. She's doing just fine. So it's really true. And what I'm going to do is I'm going to slow down what happens. As you can see, we fed her after we take blood and take wing and release her. And here she is flying. Look at that. So again, remember, she weighs the same as a lab mouse. But what we found, so bats, as they age, they seem to increase their ability to repair their DNA. They're able to remove this cellular damage. They keep this very, very stabilized and equally matched inflammatory and anti-inflammatory response to their immune system. They maintain homeostasis. And that we got to push forward. We're now right at the edge where we have the technology, we have the expertise, we have the interest, we can do things. And in the next 10 years, I really, really hope that we're going to see first drugs coming for bats. So I want to thank everybody in my lab. I have the most extraordinary group of people that I work with, and we couldn't do any of this without them. I also have extraordinary collaborators, and many of you are here in the audience. And I want to say thank you all for listening.
Well, thank you so much, Emma. That was an amazing talk, as always. And we have a few minutes for questions.
Yeah, so here.
Where's the microphone? Yeah, go ahead.
Yeah, thanks. It was very insightful. About the iPS cells, are they easy to generate or hard? And could you, for example, do reprogramming, chemical reprogramming towards rejuvenation on those cells? Are you planning that?
So, that's the first question. Are they easy to make? Now they are, because we have the Yamayaka process, but no, they weren't in the beginning. They were extremely difficult, and the only way that it actually worked, so it was the middle of lockdown. Thomas Zawaka is a regenerative stem cell biologist in Mount Sinai. These were all the people were dying, horrible stuff going on in COVID. Said, right, I want to be able to make a model, a lung model from a horseshoe bat that's resistant to COVID. And so we ended up getting, DARPA flew this bat from Spain, this dead bat in, and they thought they would be able to just use standard Yamayaka processes. No, you could not. Didn't work. But Marion de Josette, so she's a lead author in this, they had the skills to be able to realize that actually these little things they were seeing, potentially they were these stem cells. And they also modified things. So given the skill set from people working on human stem cell therapy, to be able to take that expertise to work on bats. And the other catch was that, or the, I suppose, fortuitous event, was the horseshoe bat that was caught in Spain was pregnant. So there was embryonic tissue. And so with that, they learned how to first reprogram the embryonic tissue, and then they were able to move forward. Now it's simple, yes. You can do all the tests you want, yes. Have they tried to rejuvenate it? No, we have a jet.
Okay, thanks.
All right. What have we got? Another question there?
Last year, if I remember correctly in your presentation, you said that bats, first of all, their telomeres don't shorten, and secondly, that there's even telomere elongation during hibernation.
Yeah.
You suggested that for the normal telomere maintenance, not for hibernation, but their bats don't use telomerase, but ALT, alternative lengthening of telomeres. And now this year, you said that for the hibernation, the telomere elongation that occurs during hibernation, there is indeed telomerase expression that is responsible for it. So do bats use two different mechanisms for this, or what does the evidence currently suggest?
I think we have to blame Vera for this. So we were trying to, we still have not found any telomerase in the blood transcriptomes ourselves from myotis. Working with Vera and Andre, they have found telomerase using these trap assays, which are looking at the protein that you could see in the fibroblasts. For the horseshoe bats, so these are all different species of bats. For the horseshoe bats, they've now shown evidence of telomerase during hibernation. So yes, certain species of bats are using telomerase. You can see it in these fibroblast wing cells. Is it happening in the blood? I'm not so sure. Potentially it is. But to really answer that question, we're going to have to take a different view. We need to start looking at the proteins. Are they using alternative telomere lengthening? I know Andre and Vera looked at that trap. I said, no, it doesn't look like that. So that was our speculation because they were lengthening and we couldn't find any evidence of telomerase. So if I was putting money on it now, and I'm not a betting woman, but if I was, I'd say they are. And they're using transient telomerase expression at different times. Maybe it's going on at different times, different tissues, but still they're doing this without getting cancer. So as you add more data, sometimes your conclusions change. Yeah. Hope that explained it.
There.
Yes, thank you for your presentation. I'm wondering what is known about the lifespans of megabats and how they compare to the lifespans of, let's say, more ordinary-sized bats, given that ordinary-sized bats seem to be an exception to the general correlation between physical size and longevity. So are megabats longer-lived, or do they have similar lifespans to ordinary-sized bats?
So one of those bats, so the Rosettus, the one bang smack in the middle, is a pteropodid, one of the megabats. So the big, large ones, they live longer than expected, but not as long as the little, small, temperate insectivorous ones. They hold the record. But they're still, they're living up to 20 years, 30 years, still doing much longer than would be expected. But definitely, if you want to live longest, be a temperate, insectivorous, frying bat.
Thanks, Emma. Okay, I'm afraid we have to stop now and move on to the panel, which is going to be a challenge because there's so many questions I want to ask. But could Vera and Steve please come up and take